Often I get asked what the NPK of my worm castings are, but what if I told you that pouring NPK fertilizer on your garden is kind of like trying to feed yourself with nothing but vitamin pills? Sure, you might survive for a while, but you're missing the whole feast. Here's the thing most gardeners don't realize. Your plants don't actually eat NPK. They eat what comes out of billions of living organisms in your soil. And when you understand this fundamental difference, everything about how you grow food changes. I'm Sunny with Sunnyside Soil, and today we're going deep into the soil food web, the underground ecosystem that's been feeding plants for 450 million years or so, long before humans invented synthetic fertilizers. By the end of this video, you'll understand why feeding your soil organisms is infinitely more powerful than feeding your plants directly. So let's start with what we've been taught. NPK, nitrogen, phosphorus, and potassium. Those are the three big nutrients. Walk into any garden center and that's what you'll see. 10, 10, 10, 5, 10, 5. Numbers that promise to solve all of your problems. Now, don't get me wrong, plants absolutely need these nutrients. But here's the critical misunderstanding that's been sold to gardeners for over a century. NPK is only 3 of the 17 essential elements that plants need to complete their life cycle. Plants need 3 secondary nutrients: calcium, magnesium, and sulfur, in quantities that are sometimes larger than phosphorus or potassium. Ask any tomato grower that's gotten end rot and they'll absolutely tell you that calcium is extremely important. According to research from the Mississippi State University Extension and the Mosaic Crop Nutrition, modern high-yielding crops actually remove more calcium, magnesium, and sulfur from the soil than ever before. Yet these nutrients are completely absent from most fertilizer programs. Let me give you some perspective. Calcium is the 3rd most abundant nutrient in plants. It's essential for cell wall structure, hormone regulation, and root development. Without adequate calcium, you get blossom end rot in tomatoes, hollow heart in potatoes, and weak plant stems that collapse under their own weight. Magnesium is literally the central atom in every chlorophyll molecule. That's the molecule that makes photosynthesis possible. Research published in multiple agricultural journals confirms that magnesium deficiencies prevents plants from utilizing nitrogen and phosphorus effectively, no matter how much NPK you dump on it. And sulfur? Well, sulfur is a building block of amino acids and proteins. According to Ohio State University research, plants need approximately 1 pound of sulfur for every 10 pounds of nitrogen. Yet sulfur levels in soils have plummeted since the 1980s. Then there are the 8 micronutrients: iron, manganese, zinc, copper, boron, molybdenum, chloride, and nickel. Yes, plants need these in tiny amounts as well. We're talking parts per million. But according to research from Sound Agriculture and Iowa State University, a deficiency in even one of these can completely collapse your yields. Here's why. Micronutrients are the catalyst that make everything else work. Research shows that approximately 1/4 to 1/3 of all proteins in a plant require metal ions to function. These aren't optional. They're the spark plugs of plant metabolism. Here's an example. Iron activates the enzyme that creates chlorophyll. Without it, you can dump nitrogen on your plants all day long and they'll still be yellow because they can't make the machinery to use it. Zinc is estimated to be in over 1,200 different proteins in plants and is the most commonly deficient micronutrient worldwide. It's essential for growth hormones, enzyme systems, and carbohydrate formation. Boron is the bus that carries calcium into plants. Without adequate boron, you can have plenty of calcium in your soil, but the plant can't access it. Boron is also critical for pollen germination and cell wall development. Molybdenum is required for nitrogen fixation. Plants can't convert nitrate to usable proteins without it. Manganese is essential for photosynthesis, nitrogen assimilation, and respiration. According to research from Omics Canada, molybdenum deficiency reduces nitrogen and phosphorus uptake, while zinc deficiency reduces overall vigor and the uptake of other nutrients. Now, these aren't just isolated problems. They cascade through the entire plant system. Now, here's where it gets really interesting and really complicated. Nutrients don't work in isolation. They interact with each other in ways that no NPK bag can account for. Research from Iowa State University shows that high phosphorus levels can induce deficiencies in copper, zinc, iron, and manganese. Excess potassium can trigger boron deficiencies. High calcium levels can reduce magnesium availability, and vice versa. This is why synthetic fertilizer programs often create more problems than they solve. You add high phosphorus fertilizer, which locks up the zinc. Plants become zinc deficient, which reduces their ability to use nitrogen. So you add more nitrogen, which creates more metabolic demand for magnesium. But you haven't added any magnesium, so now you have a magnesium deficiency, preventing chlorophyll formation. Do you see the spiral? But in natural ecosystems, plants don't absorb nutrients from bags. They get them from a complex network of organisms that mine, transform, and deliver nutrients in balanced ratios in exchange for something that the plant produces, exudates. Think of it this way, NPK fertilizers are like taking 3 vitamin supplements and calling it a complete diet. Quick response, sure, but zero nutritional complexity. The soil food web? Well, that's the farm-to-table meal prepared by thousands of specialized chefs, each contributing unique nutrients, enzymes, hormones, and compounds in the exact proportions and forms that plants evolved to receive. According to Dr. Elaine Ingham's soil food web research, plants produce simple sugars through photosynthesis, and they inject 30 to 40% of those sugars into the soil deliberately to feed bacteria and fungi. This isn't waste, it's an investment strategy that's been perfected over 450 million years. Plants are literally farming their own nutrient providers underground. Let me introduce you to the cast of characters living in every handful of healthy soil. At the foundation, we have bacteria and fungi, the original nutrient miners. Research published in the Soil Food Web School documentation shows that bacteria and fungi produce enzymes that break down organic matter and crystalline structures in rocks, sand, and clay particles. These parent materials contain iron, boron, phosphorus, calcium, potassium that were all locked up into forms plants can't access directly. But bacteria and fungi, well, they're the chemical wizards. They release enzymes that dissolve these structures, absorbing nutrients into their own bodies. A single gram of healthy soil can contain 4 to 5 billion bacteria and several hundred meters of fungal hyphae. Now, here's where it gets interesting. These nutrients are now locked inside the microbial bodies. How do they get to their plants? Enter the predators, Protozoa and Nematodes. These microscopic organisms graze on bacteria and fungi, well, like cattle grazing on grass. But here's the magic. Bacteria and fungi have a carbon to nitrogen ratio of 4 to 1, while bacteriovorous nematodes, they have a ratio of about 5.9 to 1, according to research from UC Davis. Well, great, Sonny, thanks for all the numbers, but what does this actually mean? When a nematode eats bacteria, it's consuming more nitrogen than it needs for its body structure. That excess nitrogen, well, it's excreted as ammonia plant-available nitrogen directly into the root zone. Research published in scientific reports have demonstrated that nematodes can contribute 8 to 19% of total nitrogen mineralization in soil annually. A study in New Phytologist found that protozoa and bacteriovorous nematodes together contribute approximately 30% of total net nitrogen mineralization in many ecosystems. But they don't just cycle nitrogen. Research from 2016 showed that the presence of nematodes increased net phosphorus availability by 23%. And this was the entire nematode community working together, not just one species. Now I can see yourself asking, but Sonny, I thought that nematodes only caused problems for gardeners. Well, those are the root-feeding nematodes. There are many different species of nematodes, including bacterial-feeding nematodes, fungi-feeding nematodes, and predatory nematodes. Now we need to talk about the most important symbiosis in terrestrial ecosystems, mycorrhizal fungi. These fungi form relationships with about 80 to 90% of all land plant species, including most of your garden vegetables. Here's how it works. The fungus grows inside and around plant roots, forming specialized structures called arbuscules, kind of like little tree-like exchange sites. Research published in PLOS ONE and multiple studies in New Phytologists show that plants can receive up to 70% of their phosphorus and more than 40% of their nitrogen through this mycorrhizal pathway. Now, in exchange, plants allocate 20 to 40% of their photosynthetically produced carbons to feed this fungus. Now, this might sound expensive, but consider what the plant gets in return. Access to phosphorus and nitrogen that would otherwise be completely unavailable, plus enhanced drought resistance, disease suppression, and connection to a common mycorrhizal network that can span entire forests. According to recent research from the University of Florida IFAS Extension, mycorrhizal fungi also secretes glomalin, a glue-like compound that binds soil particles into aggregates, literally building soil structure while extracting nutrients. And these are just the major players. There are also predatory nematodes that eat other nematodes, microarthropods, earthworms that process organic matter and stimulate microbial activity, and countless other organisms that, well, we're still discovering. Research in Frontiers in Microbiology found that these organisms don't work in isolation. They form complex co-occurrence networks where bacterial and fungal diversity interacts synergistically to enhance nutrient cycling. The complexities of these networks is directly linked to your soil health. So now that you know the players, let's talk about how this underground economy actually functions. Enter the rhizosphere, the narrow region of soil directly around roots. It's where the magic happens. According to Soil Biology Primer, this zone is teeming with bacteria that is feeding on sloughed off plant cells and the proteins and sugars released by the roots. Bacterial populations here can be 10 to 100 times higher than in bulk soil. The plant is not just a passive participant. It's actively managing this ecosystem through root exudates. Those are carbon-rich compounds that I mentioned earlier. Research shows plants produce exudates strategically, releasing specific compounds to attract specific microorganisms depending on what nutrients the plant actually needs. Here's something crucial that most gardeners don't know. There are two main energy channels in the soil food web, bacterial and fungal, and they work at different speeds. Research in Science Direct Topics shows that bacterial pathway is faster. Bacteria reproduce quickly, are consumed quickly by protozoa and nematodes, and nutrients cycle rapidly. This is ideal for annual vegetables and grasses. The fungal pathway is slower but more stable. Fungal hyphae can remain intact for soil for hundreds of years, according to the Soil Food Web School. This pathway stores more carbon, builds stable soil structure, and is dominant in perennial systems like forests and orchards. Now, most agricultural soils are bacterial dominated due to tillage, which disrupts fungal networks. But research published in Environmental Science and Technology shows that balanced systems with both pathways support the highest nutrient cycling rates. So let me walk you through what happens when you add organic matter to soil with an active food web. Within the first week or two, bacteria colonize fresh organic matter, multiplying rapidly as they consume simple sugars and carbohydrates. Weeks 2 to 4, protozoa populations explode in response to bacterial growth. As protozoa graze on bacteria, they excrete excess nitrogen as ammonia. Weeks 4 to 8, fungi begin breaking down more complex compounds, cellulose, lignin, tough plant materials. Studies show fungi produce acidic enzymes that also help dissolve mineral nutrients from soil particles. And on an ongoing timeline, Mycorrhizal fungi extend hyphal networks up to several meters from the roots, accessing nutrients and water far beyond the root zone and trading them for plant carbon. Research from PMC demonstrate that without both decomposers, that's the bacteria and the fungi, and predators, that's the protozoa and the nematodes, present, nutrient cycling cannot occur effectively. You need the whole community working together. Now, here's a bonus that most people miss. As this biological activity occurs, beneficial bacteria produce sticky compounds that glue soil particles together into microaggregates. Then, fungal hyphae bind these microaggregates into larger macroaggregates. This aggregate structure is what gives you that beautiful, crumbly soil tilth. Research in Frontiers in Microbiology, studying rice fields in southern China, found that microbial activity within different aggregate sizes directly correlates with nutrient cycling rates, and that bacterial assemblages in smaller aggregates were particularly important for accelerating soil functions. All right, I know you're thinking this is all great information, Sonny, but how do we actually feed this underground ecosystem? This is the fundamental shift in thinking. Instead of asking, what nutrients does my tomato plant need? Ask, what do the organisms need that will feed my tomato plant? The answer is organic matter, and not all organic matter is created equal. Finished compost provides a diversity of food sources. Fast reproducing bacteria will attack simple compounds while fungi work on the complex materials. Research shows that quality compost can contain billions of beneficial organisms per gram. But here's the key, it needs to be finished compost. Hot, unfinished compost is still going through rapid bacterial decomposition. Now, that's fine for soil building, but for nutrient cycling, you want that stable, complex, organic matter that feeds the whole food web. Cover crop residues, grass clippings, crop trimmings, these are all bacterial rocket fuel. High in nitrogen, easily decomposable, they'll trigger rapid bacterial growth followed by protozoan and nematode blooms. Studies on nitrogen mineralization show that adding fresh, high nitrogen plant material can increase nitrogen availability by 25% or more when nematode communities are present to recycle it. Now, worm castings deserve a special mention here, and not just because that's what I happen to sell, because earthworms process organic matter through their gut, where it's inoculated with beneficial bacteria and mixed with minerals from soil particles. The result is material that's immediately available to plants and seeded with beneficial biology. Science Direct research shows earthworms favor bacterial-dominated energy channels, accelerating nutrient cycling. Their burrowing also creates channels that increase oxygen levels, and beneficial organisms thrive in oxygen-rich environments while many pathogens are suppressed. Mulch. Surface mulch feeds fungal pathways primarily. As fungi break down woody materials, they sequester carbon for the long term and create stable soil structures. Research shows fungal-dominated soils are particularly effective at suppressing weeds because the acidic enzymes fungi produce inhibit weed seed germination. Once you've built up that soil biology, don't destroy it. Research consistently shows that tillage is devastating to fungal networks and soil structure. Every time you till, you sever fungal hyphae, you disrupt aggregate structure, you expose organic matter to rapid oxidation, and you kill earthworms and other beneficial organisms. Studies comparing tilled versus no-till systems show dramatic differences in mycorrhizal colonization and soil organic carbon levels. Now this one's critical. Remember that plants are pumping 30 to 40% of their photosynthetic carbon into the soil to feed the food web. No living roots, no food for the biology. This is why cover cropping is so powerful. Winter cover crops keep the biological pump running even when you're not growing food crops. Research shows that continuous living roots maintain higher microbial biomass and diversity. Now, if you have a plant diverse species, different root exudates feed different microbial communities. Research from End Systems studying bacterial fungal relationships found that the balance between positive and negative bacterial fungal associations is directly linked to nutrient cycling strengths. Monocultures select for narrow microbial communities. Diverse plantings create a complex, resilient food web. You could definitely add specific amendments to address deficiencies. Amendments like rock dust provides minerals that bacteria and fungi can slowly make water soluble. Kelp adds trace minerals and growth hormones. And biochar creates a habitat for microorganisms. But here's the thing, if your soil food web is functioning, many of these additions become less necessary. The biology will mine what's already in the soil. So what exactly happens when you shift from NPK thinking to food web thinking? Well, in case you haven't caught on yet, you get healthier, more resilient plants. Plants fed through mycorrhizal and biological pathways receive not just NPK, but dozens of micronutrients, enzyme hormones, and compounds that synthetic fertilizers don't provide. Research shows that mycorrhizal plants have dramatically improved stress tolerance, they can handle drought better, they resist disease more effectively, and they grow more robust root systems. And unlike synthetic fertilizers that can leach away with the first heavy rain, nutrients cycled through the food web are held in living biomass until they're needed. Studies show that this dramatically reduces nutrient loss to groundwater. That aggregate formation that I mentioned, it translates to better water infiltration, improved drainage in clay soils, better water retention in sandy soils, and reduced compaction. Research from the Soil Food Web School shows well-structured soils can reduce irrigation requirements by up to 50% just in the first year. Research indicates that fungal hyphae remaining intact for hundreds of years can sequester 20 metric tons of carbon per hectare per year in some systems. You're not just growing food here, you're actually fighting climate change. Once established, a healthy soil food web is self-sustaining. You're adding organic material that you can often produce yourself or source locally, not purchasing synthetic fertilizers shipped from who knows where across the world. And here's something studies are increasingly validating: nutrient density. Plants grown in biologically active soils absorb a more complete spectrum of nutrients. And yes, you can taste the difference. Higher Brix levels, more complex flavors, and better nutrition. Look, I get it. Walking into a garden center and grabbing a bag of 10-10-10 is easier than building compost, managing cover crops, and thinking about soil biology. But here's what I've learned through hundreds of conversations with gardeners who have made this shift. Once you see your soil as a living ecosystem rather than an inert growing medium, everything changes. Your garden becomes more than a place you extract food from. It becomes a partnership with billions of organisms that have been perfecting nutrient cycling since before dinosaurs walked the earth. The soil food web has been feeding plants for 450 million years. It works, it's resilient, it's self-regulating, and it's already in your soil right now, just waiting for you to feed it. So stop feeding your plants and start feeding your soil organisms. They'll take care of the rest. And if you want to dive deeper into this topic, I've got links in the description to research papers that I've referenced, plus resources on soil food web testing, compost making, and cover cropping. And if you're working on building your own soil biology, I'd love to hear about it in the comments. What's working for you? What challenges are you facing? Thanks for watching, and I'll see you on the next one.